So lady. So, like I said, base pairing in DNA, guanine pairs with cytosine, and it has 3 bonds. Adenine pairs with thymine, and it has 2 bonds. So, based on this, or what I explained, which one is harder to break the bond? Yes. Can you explain why? So since it's much stronger, it's gonna take like a lot of force to like pull it and break it. But with adenine and thymine, it's since it has true bond like together, it's gonna be easier versus this one. Okay, so that's DNA, and this is RNA. In RNA, uracil U replaces thymine. So whenever Laura gives you like that DNA sequence, right? If it's talking about RNA, change the T to U. U replaces the T. Base pairing in RNA is same thing, right? Guanine pairs with cytosine, 3 hydrogen bonds. Adenine pairs with uracil. Now it's U, not T. You see? The T changes to U because this is not DNA with RNA. But in DNA, it's just going to be AT. Do you see? That's DNA. And then in RNA, GC, A, and thymine will replace the U, uracil. It's the same thing. But you just replace U to T or T to U. And this is the structure of how it works. Just like first prime, second prime, third prime, fourth prime, fifth prime will be like at the top, and then phosphate group will come and join them. And then whenever a new nucleotide will come in, it's going to come join at the third prime, and then it will start forming. And this is how cytosine looks like NH2, oxygen, blah, 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 oxygen, guanine with like, if you want to visualize like 2, NH, N2. It's better if you like write it, right? Like in like a scrap paper, notebook, and try to like visualize it. Because I'm not a visualized person, so like I like to visualize it. These are the double-stranded DNA, sugar phosphate, deoxyribose.
Yeah, deoxyribose.
Yeah. And then in ribonucleic acid, it's usually single-stranded sugar phosphate. DNA has 2. RNA has 1 single nucleotide base. And it has OH on the second and third primes, while in DNA, it only has H and OH. So that's how you can distinguish which one is DNA, which one is RNA. So you can just like have like a visualized DNA double-stranded with H and OH. RNA has single-stranded nucleic base with OH and OH on second and third prime. And then in RNA, it changes from thymine to uracil when they're base pairing with each other.
So something to be said about DNA. DNA likes to be bound to itself because it's more stable.
Yeah.
Where RNA, again, doesn't have to last as long in the cell. So you'll see it floating around single-stranded, although it can bind. But it also oftentimes binds to itself.
That's why it's called single-stranded.
Yeah.
Because they like to be alone.
Can you break down like the different, like the uracil, like and what it looks like? The molecule looks like, or do we not need to know?
I believe that you do not need to know that.
So as long as you know, like, what's the difference between DNA and what, like, base pairs with each other, you should be good.
And the amount of bonds that they make.
Yes.
Those are the important components.
So GC makes 3 bonds, AU or AT, depending on what she's being asked, like DNA or RNA, has 2 bonds.
Ready? Oh, it's my turn. All right. DNA directionality. So we're getting into the directionality. How do you guys feel about being able to figure out what direction the DNA is going? Not so good? So? You feel pretty good? Okay, well, we're gonna go over it, and we'll try to work through some problems so we really feel good about it, because it's not either it can always change.
Yeah.
So strands basically run anti-parallel to each other, which means that you've got, as you can see, 5'3" prime and 5'3", and so they're going opposite directions. DNA, like I said before, prefers to be double-stranded, and RNA can base pair with other RNA, but oftentimes you just usually find it base pairing with itself. So let's see. And I talked about the stability, and we already talked about the bonds, so we'll move past that. Any question?
What they are, like action bond?
So just to be talking about these, just to be clear, these are the hydrogen bonds between the nitrogenous bases. Right. So the process of copying DNA into complementary RNA polymer. Yes.
So does the template strand always have to be 3 to 5, or can it be 5 to 3, which is like the RNA? Because RNA has to be coded 5 to 3. So does that mean the template strand always has to be 3 to 5?
So that's correct. The template strand will always be 3 to 5. That's how you identify the template strand. But oftentimes the questions will be, you need to find the template strand and then use the template strand to make the RNA. And so you just have to remember that if it's going 3 to 5, then the other one's going to be going 5 to 3. So it's just keeping that in your head and recognizing your template strand, because that's the most important step, is recognizing that template strand and then keeping the two directions in your head.
Yeah, you guys will, I don't think you guys had the discussion yet, but there's a similar question like this, which Laura kind of switches it. So just know that she's gonna give you like, her exams are gonna be similar. It's not gonna be like from the beginning to end. She's gonna give you maybe from like 3' and to like 5', and then you have to figure out where it's gonna translate, or like where is the coding strand and where is the non-coding strand, and you have to distinguish. It can always change.
So any questions on that before we move to the next slide? We can always go back if you have questions. Yes.
Also, like, so is it just like, is the DNA like a continuous strand, or is there like or is it like a chunk of DNA that the RNA polymer, like I guess how does the RNA, how does the RNA polymerase know what is a 3' and a 5', like if it's all, because
So there are going to be certain, you're getting into kind of complicated territory, but I'll give you a brief description and you can do what you will with it. So certain parts of the DNA, when the cell wants to use those parts of the DNA, they're basically opened up. And then there are going to be certain areas that the RNA polymerase will land on and then transcribe from. Yeah, transcribe from. Yes. Good. Yeah. So it's the transcription factors are the thing that tells them the RNA polymerase where to land, basically. I mean, among other things, they do a lot of other stuff. Cool. All right. So here we go. Let's get some practice. So if the top strand in this sequence is the coding strand, which way is transcription going?
oh, well, are we 5 to 3?
So top strand is the coding strand.
Oh.
All right. So that's Yeah. So then the bottom strand would be the template strand, right? And so then when you're coding, you always go in, you can either say for the template strand, you can say you're going from 3 to 5, and then so the strand that you're coding will go from 5 to 3. Is that confusing, or do I need to go over it again? Because that sounds confusing to me.
Yeah. Should we like draw it out
Here.
in the board?
So
Yeah.
Correct. Yes. Yes. So I will, let's see, which way is transcription going? So I'll just point it out on this slide. So think of it, I like to think about like rails on a train track. So you've got a little car, which is the DNA polymerase. Since this is, it said this was the coding. It's gonna ride the car, it's gonna hop here, hop on right here. And as it rides, it's gonna create the RNA strand. So it's gonna go this way along here, and it's actually going to create this strand. So the coding strand is the strand that's going to get turned into the DNA. But what happens is both these are into RNA. Both these are DNA strands. So these will actually technically get separated. The RNA polymerase will come in here. It will land on wherever the transcription factor tells it to. And then it's going to run this way, and it's going to make this here, except for, again, the T's will all become the U's. Does that help to clarify that? Yes. Correct. Yeah, so let's see, this is again 3 to 5. So because we've identified that this is the coding strand, then we know that this has to be the template strand. And so you always imagine your car, your RNA polymerase on the template strand. And your RNA polymerase has to always go from 3 to 5, right? But then when it goes 3 to 5, because the strands are anti-parallel, then it transcribes a new strand from 5 to 3. Does that make sense? Yes.
So the coding strand is different from the template.
Correct. So the coding strand relates to actually just DNA. There is no, in RNA, there's no coding strand from what I understand. It's just when you're looking, because right now we're looking at 2 DNA strands, because notice there are no U's where the T's are.
Yeah.
Otherwise, that would be an RNA strand.
So I feel like it's a weird name. That's what I want.
Well, so the reason for that is during DNA replication, the DNA comes in, and then so you have a DNA template strand, and then they're coding another DNA basically into it. So that's why during, mitosis and meiosis. Yeah. So do we feel comfortable with that? This is a complicated, yes.
Is the coding strand the same as the non-template strand?
Yes.
It's like literally the exact same thing, or just the same?
Same thing.
It's almost exactly the same, except for that if we're transcribing, then all the T's will turn to U's. But besides that, it's the same.
Mm-hmm.
Does that clarify that, or do you want me to?
Yeah, I think that.
Yes.
So since the bottom one is the template strand,
Mhm.
does that mean that the RNA is coding backwards to make it 5 to 3? Like it's being, that nucleotides are being added backwards?
So they're from the slides that we showed before, they're being formed in the exact same way as we explained before. They're always formed in that direction. Because remember the phosphodiester bond and all that kind of stuff. So that's what's going on when this is happening. It's just that it's starting here. Well, I'll give you another example. If we were, let's just say now, because erroneously identified this as the coding strand. It's just a choice. Because really, RNA polymerase can come in and code off of either one of these. It just depends on where the cell tells it to go. So we could say that now this is the template strand. I can just make that up, top one. And so then if this is the template strand, the RNA polymerase is going to bind here, and you're going to get, this is going to become the RNA strand right here. So you're going to get C, C, G, G, U, C, A, U. Cool? Yes.
So for the question, when it asks which way is transcription going,
Correct.
if the top strand is coding, it would be going to the left then, right?
From here, top strand is coding. Yes.
Yeah.
Yep. Yes.
Is she gonna tell us which one is the coding strand and which one is the strand? Like, if they're-
She has to,
Yeah.
because there's no way for you to tell
Oh.
if there isn't. She has to tell you either one's the coding strand or the template strand, or she's going to tell you where the transcription factors are, or where there's a promoter. She has to give you some indication. What's
like this way, like the question, because it's going this way.
Mhm.
Can we write down that it's just like mirroring, like inverted, or is there like a certain word that we have to?
there isn't necessarily a certain word that you have to use. I mean, you could say that they're technically, you could say it's anti-parallel.
Yeah.
That's the technical term. And no, that's probably, if you want to use a word, anti-parallel is the word.
Yeah.
Yes.
So since RNA always encodes 5 to 3,
Mhm.
it just goes different directions depending, transcription happens at different directions depending on how the template strand is.
Correct. Yes, and that's what makes it confusing is, you could get either one being the template strand, and that's where people get thrown off, because you can't just say it's always going left or it's always going right. Yes.
So does that mean the RNA would enter the RNA polymerase from a different entrance, or?
So all of this is floating around inside of the nucleus. And so it's basically just finding the transcription factor that it's going to land on. And there's an attraction between them, like kind of like a bond. Basically, there's an attraction between there's an attraction between them, and that's how it's found. They're just literally floating around.
So another suggestion, I know this topic is really confusing, even for me, like sometimes I get hard time, right? So like, even when I was in this class, I was like struggling to understand best way that like, if you guys still are confused with this part, try to watch YouTube videos. There's like some professors or some people that could explain it like so much better than we might not be able to. And maybe like, sometimes it's hard whenever like the way that some people teaches, like you don't understand. So like trying to find different videos related to whatever topic that you're struggling on, and they will just like break it down. I know there's like a video that like I watched when I was when I took this class, and I was like, okay, so it just makes sense. Just watch it, go over it, and then try to understand what direction they're moving.
More slides about transcription. How exciting. Okay, so here. Okay, so this goes a little bit into what we were talking about before, just the promoter, like you guys were asking, this is one of the things that you would tell you, okay, the promoter's here. So then you know that if the promoter's here, if the promoter's here, this one has to be the template strand, and this one has to be the coding strand, or technically. So template strand is on the bottom, and this will be the new line of RNA. And actually, it explains, this is actually what I was talking about before. So it goes in, and it kind of pinches it and then codes it from there, and it'll come out, and then you end up with this strand here. Yes.
Wait, I forgot my question. Sorry.
If you need to go back, just if you remember it, we'll go back and figure it out.
If you look here, right? These are anti-parallel. And then when the RNA comes, see how these are same, like 5' to 3', or 3', I mean, sorry, 5' to 5', and then 3' to 3', but the T is gonna change to U, right?
Mhm.
Because it's RNA.
Mhm.
And these are interparallel, and then this 5' will come in, they're the same, but it just changes the T to U.
Okay, so, the DNA is unbound at the promoter site to be read?
Yes.
Okay, so at the promoter, that's where, unwinds to be read. Okay.
Yes. And so basically, that's what this is an example of right here. It's just showing you basically because it shows that it's starting you a promoter, but it has this kind of opening that you would see here. And then the RNA polymerase can pop in there, work along, because it's going to be working along the bottom strand here. And then it'll basically pump out this thing right here.
And then it opens up, and then the RNA will come
Yeah.
in. Slide it through.
Yep. And then it's done and it gets knocked off at the terminator in some place.
And then that's the completed RNA transfer.
Yep. Cool. We good?
Yeah.
Do you have another?
No.
Okay. Great. Okay. Okay, so we'll go over this, I guess. So this first part, promoter start point, that's the initiation phase. And so again, the transcription factors, the RNA polymerase binds to the promoter region of the DNA, as we've kind of discussed before. This will just kind of lay it down straight form. So, and then we have the elongation phase, which is basically, so you see it, it's landed, it started, it's going to move along here. So this is the elongation phase all the way through here. And then you get termination, which is when it hits the termination area. So there, actually right there, the RNA polymerase is knocked off. There are different kinds of termination, which you guys don't need to know. Maybe, yep, which is great. Yes.
So
I believe, well, that is, the terminator can be different things. Sometimes it's an actual, I don't know how much I want to go into it because you don't technically need to know, but it could be, I'll just tell you, it could be a loop. Sometimes the DNA will, or the DNA strand will bind to itself, and then it'll cause the RNA polymerase to knock into it and it'll come off. Or there could be something attached to the RNA polymerase itself that will knock it off. So there are different instances, yes.
it's like a sequence of nucleotides.
Correct.
So like the TATA sequence or
Mhm.
CAAT sequence. And that sequence is what tells the RNA polymerase which direction to go.
Yes, and there are the transcription factors play a part in that. The transcription factors do different things. They will help the DNA polymerase bind better to the promoter. So they actually need, the transcription factors are very necessary to that. So it's if I remember correctly, it's actually the transcription factor that binds to the promoter first, and then it helps the RNA polymerase to land, and then there are other transcription factors that kind of set it ready to go. So think about like a plane lifting off. You've got all these other people working with it to get it to go.
What exactly is a transcription? Like what kind of visualize?
Think of it like a different, like a kind of molecule. Yeah, it's like a little molecule. Some of them are like RNA polymerase actually has two components. I think she might have actually showed you this. Like, but it's got those two components. But transcription factors can be lots of different kind of molecules like that.
Yeah, so
basically.
transcription factor can have like the start site and the stop site. Transcription start and transcription stop. Like, do you know what I'm saying? Like, you know, like transcription start site and then the promoter.
The termination.
Yeah,
site,
and
something
then transcription stop site.
Mhm.
I'm learning about that in my bio class. So like those three major thing in RNA. Yeah.
Cool.
Transcription factor or factors
Mhm.
bind to the promoter.
Mhm.
And then get that RNA polymerase in there
Yes.
and then tell it which way to go.
Mhm.
Yeah.
So I'm going to get to you in a second. I want to add a little bit more to that. The reason the promoters are needed is because the sequence, RNA polymerase can bind to a lot of different promoter sequences, but because it's not specific, like it doesn't bind to specific ones. What happens is the binding isn't very good. And so those specific promoters will bind to it and they have better binding and it keeps it on track. Yeah. Okay, so what were you gonna ask?
There were two, like, both strands of the DNA. And then obviously the transcription factors that direct the RNA polymerase to which to bind. Is there like an instance where like the transcription factors can go? I know it's based on directionality,
Yes.
but could it bind possibly to like this DNA strand first, then notices it's not the right direction to the other?
So
Is it like
the way it works is it's all about Rutsch's it's complementary base pair. And so it won't want to bind somewhere that it doesn't bind well. And so in this case, I'm actually not sure that I think this is a little oversimplified because really the RNA polymerase can only bind on one of those because it won't bind on the other one because it can't synthesize in that direction.
I'm thinking of it in terms of like, when the nucleotides are added and notices that it doesn't bind, doesn't fit, so then it gets like,
Oh, so
great, like that concept. Is
Okay.
it kind of?
so I might not have understood what you're asking, but I'm going to say this and you can tell me. What you're asking. So are you talking about like the wrong nucleotide getting bound to in the sequence of nucleotides? Is that what you're talking about? Or
More so just like before transcription actually begins and that RNA polymerase binds to that
Mhm.
promoter region.
Mhm.
That all has to happen until the transcription factors actually bind to that.
Mhm.
But then way before that, could there be an instance where it binds to person like the wrong region? And then notices that it's bonded wrong and just puts it back to the right one, or is it just straight?
basically inside the cell there are ridiculous amounts of strands of DNA, and what actually prevents them from binding to the wrong is that they're wrapped around histones.
Yeah, you're gonna learn about that later on. You will learn about histones in like chapter 10.
So and that basically just prevents them because there's no access.
Oh,
Yeah.
yeah, that was just.
So you'll learn more about this at the end. This is just the basic underline.
But it's good that
yeah.
good to ask questions. Encourage that, yes. No, not that I know of. If she introduces one, but she hasn't discussed anything. So that was all like what I told you about. It was upper level course stuff. So although she does get in there sometimes.
Maybe take next class with her.
Okay. So RNA polymerase transcribes times the introns are spliced out. Okay. So we saw the transcription. I see. So I'm just going to go back here. So this is taking place after the last step here on this one. So once we get the completed RNA transcript, then we're moving on to this one. And that is right here, I believe. And so you see RNA polymerase transcribes the gene, and then these other processes begin to happen, which was some of the stuff she discussed in class.
And then introns will leave, exons will stay, and that's what mRNA are made from. Because if you can see, right, in this region, see, these are the exons and these are the introns. And then this is the protein coding sequence where it's going to be made into proteins. Only the red parts are in, the yellow parts are out because introns don't stay in, only exons do.
Okay, and so also we have the untranscribed regions. That's what UTR stands for, is untranscribed region. And then we have the 5' cap and the poly A tail. And this is actually the process moving from the pre-RNA, which again is what came out, to mRNA. Or so MR just means messenger RNA. So that's the process of moving through this. So you get your splicing, which you guys have gone over that process, and I think we're gonna get into it on some of the next
So
slides.
splicing is when
We'll
introns are.
We'll get onto it. I think that we have some slides about it. Yeah. So before I move on, feel good about that? Yes,
What is that?
the
Like,
splicing.
That's. Why?
Sure. So the reasons I know for it is, is that word, the differential, I need to look at the slides. We don't have one on it. Basically, these have components that are needed earlier in the process, and then they're cut out. And so you can have, I'm not going to go too far into it because I don't know the full thing, but basically these give the RNA the capacity, or the cell the capacity, to basically rearrange the exons, and that allows them to produce different cells. And so depending on, because remember, this is just a very short example, these are extremely long. So when the cell cuts these things out, they can take out, maybe they decide not to use this one here, and then they're using these two. Now they can't, put this one over here, anything like that, but you can cut that out. And how that relates to the cell as a whole is that allows you to make a skin cell, for instance, could maybe have this one cut out, and then those two would make a skin cell, and maybe all three make a liver cell. I'm just giving you examples. That's kind of unrealistic, but it's basically, yes. That's the word I was looking for. Thank you. I was like, what is that word? It's not off the top of my head, and I didn't write it down. And so she didn't go a lot into the purpose of these, and I personally, off the top of my head, don't remember a lot about that.
She's gonna talk about it in later on topics.
Yeah.
So we don't need to know it.
Yeah, for now, you don't need to know it. Yeah, and it is good, you build that big picture and things fit together and it makes sense, and you're like, it's easier.
It's basically just there for the. Like, they don't really have like a purpose except for.
They definitely have a purpose. I just don't remember what the purpose is off the top of my head, if I'm being very honest. Okay. Any more questions before I move to the next slide? Cool. All right, splicing. So, like we said before, certain introns are selected. Well, the introns are all spliced out before, or in pre-mRNA. Move to mRNA, the introns have to be spliced out. Now, mistakes do happen, and it's actually not good. Those cells will be degraded. But basically, the spliceosome is a protein. I remember she talked about this some in class. What was it? I think that was on Monday. She talked about the spliceosome a bit. But basically, it pinches the intron together, and then it cuts it out. And then you're left with an entire DNA strand. Yeah. So, and then those are called SNRNPs, small nuclear ribonucleic protein. Nuclear protein.
So in the splicing of the RNA, you can see the exons stay in that process, right? And then the yellow part, which is the intron leaves. And it forms that larget shape. It just degrades. And then the mature RNA is where you're gonna use into protein.
And again, this is a type of ribozyme. I love that word, ribozyme. So what is SNRP made of? I actually don't know.
Is that a protein that was made through transcription translation as a function of whether now? Like, is that a protein that was made? Oh.
Yep.
To use for splicing?
So all of these things are within the cell that we're actually working with them. Most of them, well, all of them are made through the same process. It's just some are RNA, some don't make it into a protein. So the ribozyme is a protein and an RNA. It's a mixture of the two. Cool. All right, I'm going to